Hello, my geniuses. Welcome to another biochemistry lecture series today. It's your great scientist Lorenzo, and we'll be talking about gluconeogenesis, which is the formation of glucose from non-carbohydrate source. Because the carbohydrate source where we can form glucose is glycogeneolysis. We have talked about it in our former videos. Well, now, how can we form glucose from non-carbohydrate sources during fasting starvation, where you have not eaten anything? How can you also form glucose from the non-stored glucose? The non-stored glucose is through glycogeneolysis, which we have talked about in our last video. But this one is what we call gluconeogenesis. So how can we form glucose from non-carbohydrate sources? So if you have watched all those videos, you can come and continue from here. So follow me to the end of the video. If you are a newcomer joining us, make sure to like, subscribe and share all these videos to other people that will need it. It's very, very important. All these things are colored pictures that you like and you use and you enjoy. Just join us. If you want the PDF, you make sure to join us and become one of us, and you always get the pictures of all these things. Very easy for you to articulate. You get it now. So also join all our social media on MedGenius Academy. So all our social media, TikTok, Facebook, Instagram, everything on MedGenius World Academy. So since you have listened and you are still here up till now, make sure to subscribe, but don't forget. So let's jump right into the video. So the first one, in the introduction to gluconeogenesis, first of all, you ask yourself, what is gluconeogenesis? What is gluconeogenesis? From the name glucose, genesis, formation. Gluconeogenesis, the formation of glucose. Gluconeogenesis, the formation of glucose from non-carbohydrate sources. You form glucose from non-carbohydrate sources is called gluconeogenesis. Formation of glucose from non-carbohydrate sources. Don't forget. So now, what is gluconeogenesis? This is a metabolic pathway which induce or involve the formation of glucose from non-carbohydrate sources, like you lactate, amino acid, glycerol, propionic acid. So you have lactate, amino acid. Which part of amino acid? We have call it gluconeogenic amino acid or glycogenic amino acid. So from lactate, glycogenic amino acid, glycerol, and propionic acid, which is propionic acid is an all-chain fatty acid. So when you are forming glucose, you are not forming glucose from the stored glycogen, you are forming glucose from the non-carbohydrate source, which is other sources in your body that is not glycogen. That's what we call gluconeogenesis. But when you are forming glucose from glycogen, you call it glycogeneolysis. Lysis means breaking down of glycogen to give you glucose. Hope you are understanding it now. Gluconeogenesis, formation of glucose from non-carbohydrate sources. What are the carbohydrate sources? Lactate, glucogenic amino acid. You have what we call propionic acid, is an all-chain fatty acid, and glycerol. So where does this thing occur? It occurs in the liver, which is the major site, and the kidney, which is the minor site. Liver, which is the major site, and the kidney, which is the minor site. Are you getting it now? And we have what we call the subcellular location, which is mitochondria and cytosol. So some works in the mitochondria, some works in the cytosol. So key idea, always know gluconeogenesis, formation of what? Glucose. Don't forget. So since you have understood it now, what is the stimulating factor? Remember, I always give you stimulating factor. What makes sure gluconeogenesis occur? How will your body look at and say, okay, it's now high time I start forming glucose? Remember, when you eat, your body cannot form glucose. Your body help you store the glucose you are eating. So you don't have to form glucose when you are already feeding on glucose, which is carbohydrate. So this one now, your body has not seen glucose. You have not finished all your glycogen already. You have broken down all. So the first to go every time you have not eaten, and when you are tired or when you are stressed, the first component to go is glycogen. So glycogen will be converted to glucose. So when you have exhausted all your glycogen, do you know where your body will focus on? Your body will start focusing on the non-carbohydrate sources. The non-carbohydrate sources like lactate and co. Your body will start focusing on them, start breaking them down in excessive amount. And as your body is breaking them down in excessive amount, it's very, very bad, because you know what? Time will reach, you start having what they call muscle wasting. You exhaust all your fat, and when you exhaust all your fat, your insulation will reduce, and it's very crazy. So your body start breaking down amino acid, your body start breaking fat very well, and it's very, very crazy. So now remember, fasting, starvation, strenuous exercise, this condition induces and causes what we call glyconeogenesis. Your body starts inducing all this condition when you are stressed, when you are tired, when you have not eaten anything. In a very stressful condition, your body starts doing gluconeogenesis. Also, remember, when all these conditions start happening, glucagon is released from the alpha cells of Langerhans of the pancreas. Cortisol is also released. Epi and non-epi are released. These are stress hormones, fight or flight hormone. Your body releases them immediately. Now, because your body needs glucose, if you like, your body needs glucose in this bad hot weather condition. Your body needs glucose. So remember, what inhibits it? Inhibiting factor when you eat. Fed state, rubbish. During fed state, your body do not want gluconeogenesis, because your body is storing the glucose, so your body cannot break them down, so it's not possible. So this one out. Also, in presence of insulin. When you eat, immediately your body releases insulin, bind to GI receptor, immediately help you store the glucose. So insulin cannot induce gluconeogenesis. Also, tyrosine too is also released to increase the basal metabolic rate. So you get it now. So in the inhibiting factor, which is well fed state, insulin, tyrosine, your body already has glucose, so your body do not need to break down glucose. Your body do not need to form glucose because it already has glucose. But in the stimulating factor, your body needs glucose and do not have glucose, so your body has to look for other sources where it can get glucose from. Get it now? So in this gluconeogenesis, your body is not getting it from glycogen. Your body is getting it from other carbohydrate sources, other sources that are not just carbohydrate. I get it now. So key nature, you have to know fasting and starvation is equal to gluconeogenesis, while fed state is not equal to gluconeogenesis. So fed state offs or inhibit gluconeogenesis. So since we have studied this part now, so the next slide is the sources of glucose. So where can you source glucose? Let's say we are not taking it from glycogen. Where are places you can source glucose from? Number one, from lactate. So from lactate, you remember Cori cycle. Cori cycle is just like a hemolytic pathway whereby your body can derive glucose from lactate. Remember, in your step 11 and anaerobic glycolysis, what do your body do? In your step 11, during intense exercise, your body oxygen level drops, and when your body's oxygen level drops, instead of going from pyruvate to acetyl-CoA into the mitochondria, your body now channels it to step 11 to form lactate. So there will be reduction from what? Pyruvate to lactate. Immediately there will be reduction from pyruvate to lactate, which is step 11, anaerobic glycolysis. In the absence of oxygen during fasting, starvation, strenuous exercise, your body channels pyruvate to lactate by what? Reduction. So when your body channels pyruvate to lactate in that high amount, if you continue playing ball, or you continue trekking, or you continue doing that thing that is strenuous for you, your body will now build up that lactate, or we call it lactic acid. Your body will build that lactate up, and when your body builds that lactate up, what will happen? It will now cause what we call lactic acidosis. In lactic acidosis, what will happen? Your lactic acid level in your muscle will increase. When your lactic acid in your muscle increase, it will start causing what? Cramps. And this is also as a result of low oxygen level. Oxygen level has dropped, you're dehydrated, you're tired, lactic acid level will increase, causing lactic acidosis. Then what does the body do? How does the body save you? Because now you already have high amount of lactic acid. What will the body do at this point? Your body will not channel that lactic acid through the blood to the liver. So it's already in your muscle. It's in your muscle. Your body will not say, hey, your lactate level is not high. Let me channel all this lactate to the liver. So when your body channels to lactate to the liver using Cori cycle, remember, glucose to pyruvate, 10 steps of glycolysis. At low oxygen level, what will your body do? Your body will now reduce pyruvate to form lactate by what we call lactate dehydrogenase, oxidizing NADH to NAD+. So that's what your body will do. And if your body do that immediately, your lactic level will increase. Because why? You are still inducing stress, you are still inducing wear and tear of your muscle. Your muscle is working too hard and there's no water there, the oxygen level have dropped. So immediately your lactate, you start feeling muscle cramps, weakness, difficulty in movement. Your body will say no more. maybe when you rested, maybe you are not hydrated, your body will not say no more. You take that lactate, pass it through the blood to the what? To the liver. When it takes it from the blood to the liver, immediately it will now go. There will now be what we call oxidation from lactate to pyruvate. There will be oxidation from lactate to pyruvate, and pyruvate will now will now bypass so many pathways, which we will say at the last part. Pyruvate will bypass a lot of enzyme in order to form glucose. Okay, now so we'll talk about the bypass in the later part of the video. So if glucose is formed now, what happens? Then you now get glucose from lactate to what we call oxidation. But at low amount of oxygen, dehydration, walking, stress, strenuous exercise, fasting, your body reduces pyruvate to lactate. But now to make you go back to normal, your body oxidize lactate to pyruvate. You get it now? So we have lactate, pyruvate, glucose. That's how we form glucose to what? Lactate. I get it now. So we call this what? Anaerobic glycolysis. And this works for anaerobic condition when the oxygen level is dropped. The next one is what we call 2-gluconogenic amino acid. So what is gluconogenic amino acid? Gluconogenic amino acid are amino acid that you can transaminate to give you what? Intermediate of what? Glycolysis and TCA. So when you break them down, when you degrade any of these amino acids, they will give you what we call intermediate of glycolysis, where you can see from glucose and you can see from ATP. If they ask you gluconogenic amino acid are amino acid that when they degrade them, they can form glucose. So the usual one they use here is what we call alanine. So alanine is an amino acid that is used in this case very well. So when you transaminate alanine now, transamination is a transfer of amino group from an amino acid to a keto acid. So go watch my video on transamination. So if you convert if you transfer amino group from amino acid to a keto acid is usually alpha ketoglutarate. So if you transfer amino acid from alanine to alpha ketoglutarate, so alanine is the amino acid, alpha ketoglutarate is the keto acid. So if you convert it now, transfer alanine, if you transfer amino acid from alanine to alpha ketoglutarate, it will give you now that alanine will become what we call pyruvate, which is a keto acid, a new keto acid. Then where alpha ketoglutarate will always give you glutamate. Don't forget. Transamination is the transfer of amino group from an amino acid, which is alanine, to a keto acid, which is alpha ketoglutarate, to form a new amino acid, which is glutamate, and a new keto acid, which is pyruvate. So that's why we call it glucose-alanine cycle. So glucose-alanine cycle is the formation of glucose from alanine, but to pyruvate. So you must form pyruvate first and bypass to form glucose. You get it now? So this is how we can get it to what we call glucose-alanine cycle. But you can also use oxaloacetate too. So if you are using aspartate, you transaminate aspartate with alpha ketoglutarate. You can also get oxaloacetate and also still form glucose. You get it now? But go watch my transamination video. You understand it there. Now we have said the first one, which is from lactate to Cori cycle. We have said glucose-alanine cycle, which is glycogenic amino acid. There are all many glycogenic amino acid though. It's not only this one. No. There's aspartate though. There are so many. There's valine. There are too many. So not only this one. So the third one is 2-propionic acid. Again, 2-propionic acid. The third one, propionic acid. So propionic acid is an odd-chain fatty acid. Odd-chain fatty acid, which is carbon 3. Propionic acid. So when you use propionic acid, so go watch my video on beta oxidation. We have beta oxidation of long chain fatty acid, of very long chain fatty acid, medium chain fatty acid, odd chain fatty acid. So when you go on odd chain fatty acid, immediately, first of all, propionic acid is condensed with acetyl-CoA. So when you condense it with acetyl-CoA, it will form propionic CoA, like you are seeing on your screen. First of all, propionic, which is propionic acid, which is an odd chain fatty acid, is first of all condensed with acetyl-CoA to form propionic CoA. From propionic CoA, it undergoes what we call series of reactions to form what? Methylmalonic CoA. You have the L form and you have the D form before you form succinyl-CoA. So the only time, remember, we said that when you break down fatty acid, you get intermediate of TCA or glycolysis. So through propionic-CoA, you can undergo beta oxidation to form what we call succinyl-CoA. So when you form succinyl-CoA, what happens? You can now undergo the reaction to form glucose by bypassing two. So you go watch my video on the all-chain fatty acid. So one thing you have to know, you can use propionic-CoA to form succinyl-CoA. From succinyl-CoA, you bypass to get glucose. Simple. So you can obtain it from fatty acid by converting it to succinyl-CoA, enter the TCA cycle, then form glucose to gluconeogenesis by bypassing two. So now in this reaction, now you require vitamin B12. So in this reaction, or using propionic-CoA, you require vitamin B12 and you require biotin too. You require biotin and vitamin B12. Don't forget. But in transamination, you require what we call vitamin B6, which is pyridoxal phosphate. So pyridoxine is also used in gluconeogenic amino acid during transamination. You get it now? Why vitamin B12 is used here, and B7, which is biotin too, is used in this beta oxidation of all-chain fatty acid. Glycerol now. Glycerol is another one. Glycerol is a fat, is also a part of a fatty acid too. Are you getting now? But it's not a fatty acid. Glycerol forms a component of fatty acid, but because you can form TAG, you need glycerol and fatty acid. So we have used the fatty acid we use here is propionic acid, which is also a fatty acid, but an all-chain fatty acid. You get it now? While glycerol is also used to form triacylglycerol, but from glycerol now, what happened now? From glycerol, you can form glycerol 3-phosphate. From glycerol, you can form glycerol 3-phosphate by glycerol kinase. So when you form glycerol 3-phosphate by glycerol kinase, then from glycerol 3-phosphate, you can use a dehydrogenase reaction to get dihydrogen acetone phosphate. You get it now? From glycerol 3-phosphate, you can now use an enzyme called glycerol 3-phosphate dehydrogenase. Are you getting now? To form what? Dihydroxyacetone phosphate, which is formed by the cleavage of fructose 1,6-biphosphate. So when you cleave fructose 1,6-biphosphate by hydrolase A, you get glyceraldehyde 3-phosphate and dihydrogen acetone phosphate. The hydrogen acetone phosphate can enter to form glycerol. Also, when you need glucose now, you can also use that glycerol that forms your fats, glycerol that forms your lipid, that forms your TAG. You can also break down that glycerol by first of all phosphorylating them at carbon 3 to form glycerol 3-phosphate, then reducing them using what we call dehydrogenase to form the hydroxyacetone phosphate. From there, you can bypass some pathways to get to glucose, which we'll talk about in the ending part of the video. So stick to the ending part. So all these things occurs in the liver and the kidneys. So since we have seen that one now, so how do we bypass this reaction? You get it now? So how do we bypass this reaction? Remember that it's very, very important. You have to know that all these things occurs in the liver and the what? Kidneys now. And you need ATP, you need biotin, which is vitamin B7, and we need CO2. You get it now? So first of all, how can you bypass? How can you start the reaction? First of all, how can you start the reaction? First of all, you have to do what? You have to add CO2 now. You have to add CO2, which is carboxylation now. You have to add CO2 to pyruvate. So when you add CO2 to pyruvate, what will you get? Adding CO2 to pyruvate by what we call pyruvate carboxylase, you will now form what we call oxaloacetate. So pyruvate carboxylase carboxylate pyruvate to form oxaloacetate. So I am bypassing what we call acetyl-CoA. Instead of forming acetyl-CoA, I only form acetyl-CoA when I have high amount of glucose I am breaking. But this one I don't have glucose, so I have to save my glucose by carboxylating pyruvate to form oxaloacetate. So when you form oxaloacetate, what happens? Now the oxaloacetate you already have now, what will you do? So oxaloacetate cannot leave the mitochondria. It cannot leave the mitochondria. You can see the oxaloacetate cannot leave the mitochondrial membrane, so it has to convert to what? Malate. You get it now? The oxaloacetate cannot leave the mitochondria. It has to convert to what? Malate. And when it converts to malate, what happens? Now malate has a shuttle system just like citrate. So malate has a shuttle system just like citrate. Citrate can also leave the mitochondria. Malate can leave the mitochondria. So for you to be able to leave the mitochondria in a TCA environment, you have to convert to either citrate or malate because they have shuttle system. So after pyruvate is carboxylated to oxaloacetate. Oxaloacetate cannot leave, so oxaloacetate is now reduced to malate, because first of all, malate, are you getting now? Malate oxidizes to form oxaloacetate. Are you getting now? So when we are now going back to malate, we have to what? Reduce. So when you reduce oxaloacetate, you form malate. And when you form malate, malate uses the shuttle system to break out. Are you getting now? To break out into the cytoplasm. So we have oxaloacetate in the mitochondria, convert to malate. Malate uses the shuttle system, go into the cytosol and form what? And go into the cytosol and reconvert back to oxaloacetate. Do you get it now? Do you get it now? So first of all, inside the mitochondria, pyruvate is being carboxylated with pyruvate carboxylase to form oxaloacetate. Oxaloacetate is a TCA substrate. I get it. It's a TCA substrate inside the mitochondria. So when it's inside the mitochondria, it cannot leave the mitochondria, so it has to be reduced to malate. The malate leaves the shuttle system, so it leaves to the cytoplasm, reconvert back to what? Oxaloacetate. When it reconvert back to oxaloacetate now, then oxaloacetate does something now. Oxaloacetate now is now converted to what? Phosphoenolpyruvate. Oxaloacetate is converted to phosphoenolpyruvate. Are you getting now? So when oxaloacetate is converted to phosphoenolpyruvate, you by what we call phosphoenolpyruvate carboxykinase. Don't forget, phosphoenolpyruvate carboxykinase. That is what oxaloacetate is converted to. So when oxaloacetate is converted to phosphoenolpyruvate carboxykinase, so when it's converted to that, it makes it easier for it to do what? Enter into the step 8. So instead of, we are bypassing what we call pyruvate kinase. Remember, phosphoenolpyruvate is converted to what? Pyruvate by pyruvate kinase. Are you getting now? But this one we go from pyruvate to oxaloacetate, oxaloacetate to malate, malate leaves, convert back to oxaloacetate, oxaloacetate convert to PEP, which is phosphoenolpyruvate. So when you form phosphoenolpyruvate, are you getting that? By phosphoenolpyruvate carboxykinase, we call it PEPCK. So when you form phosphoenolpyruvate, what happens? Then you now start bypassing people. So who is the first person you bypass? You bypass a lot of people in order to get glucose. So you bypass from step 7, step 6, step 5, step 4. Are you getting now? You bypass all this reaction and you go to what we call fructose 1,6-bisphosphate. So you are going to fructose 1,6-bisphosphate, which is the fourth substrate in the glycolysis. So you bypass from step 7 to like step 5. You leave them away. You go from there. So when you form phosphoenolpyruvate, you go down to fructose 1,6-bisphosphate. I get him now. So when you go to fructose 1,6-bisphosphate, fructose 1,6-bisphosphate will now use fructose 1,6-bisphosphatase to form fructose 6-phosphate. You get it now? Don't forget. So I told you that after you have formed phosphoenolpyruvate, I get him now, you bypassed step 10, which is pyruvate kinase. The enzymes are inhibited at this particular point. They are not useful. The enzymes are inhibited. All those 1, 3 and 10 rate limiting enzyme in glycolysis are bypassed. So step 1 in glycolysis is hexokinase glucokinase, not useful in this case. I get him now. The irreversible step of glycolysis. Step 3, which is phosphofructokinase 1, is also an irreversible enzyme of glycolysis. So you are skipping all these people, and the last one is what? Pyruvate kinase, which is step 10. So step 1, step 3 and step 10. Now these are irreversible step of glycolysis, which you are seeing on your screen. So which now I say that I'll bypass step 7, step 6, step 5, and I'll go to step 4. Now in that step 4, I'll use what we call fructose 1,6-bisphosphatase to convert fructose 1,6-phosphate to fructose 6. So I'm using phosphatase to remove phosphate at carbon 1 to form fructose 6-phosphate. That's very, very simple. I get it. I'm bypassing so many people. I bypass from pyruvate to acetic OA. I bypass that reaction. I get him now, and I go to formation of oxaloacetate. Also, I bypass pyruvate kinase to form what? PEP, phosphoenolpyruvate. Then I bypass step 7, step 6, and step 5 to form fructose 1,6-bisphosphate. But when I form fructose 1,6-bisphosphate, also, what will I do again? I will also bypass step 2. I don't need step 2 here, which is if I'm coming down, it is phosphohexoisomerase, so I will bypass that one. I don't need it here too. I will not go to step 1. I get it now. So the rate limiting enzyme here, I'm bypassing about 4 enzymes. I'm bypassing step 1. I get him now. That is a good bypass enzyme, which is glucose-6-phosphatase. I also have fructose 1,6-bisphosphatase, pyruvate carboxylase. I get him now. These are enzymes that are very, very important. These are enzymes that are very, very important. So immediately I bypass step 2, I'll go to formation of glucose. So how can I form glucose? Glucose-6-phosphate now will now form glucose to what we call glucose-6-phosphatase. Don't forget. Pyruvate to oxaloacetate, oxaloacetate to malate. Malate leaves the mitochondria and convert back to oxaloacetate. Oxaloacetate form PEP, which is phosphoenolpyruvate. Phosphoenolpyruvate skip step 7 to step 5 and form fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate undergoes phosphatase reaction to form fructose 6. Fructose 6 skip step 2 and form what? Fructose, and form glucose 6 now. Glucose 6 now use what we call glucose-6-phosphatase to form glucose. So that is how we form what? Glucose to pyruvate. You get it now, from non-carbohydrate what? Source. So the good bypass enzyme here is glucose-6-phosphatase, fructose 1,6-bisphosphatase. I get them now, pyruvate carboxylase reaction, and we have what we call malic enzymes. In malic enzymes, what happens? Malic enzyme is the conversion of malate directly to pyruvate. Malate directly to pyruvate. You use malic enzyme to convert malate to directly to pyruvate. This enzyme is very important in fatty acid synthesis. So in fatty acid synthesis, you have to form what we call malate. After citrate has left, when citrate leaves, you also form oxaloacetate. Oxaloacetate form malate. Malate form pyruvate. So from pyruvate, you can also still do the same for malate directly. You get it now? From malate directly. So these are the bypass enzymes, which are very, very important. They are the bypass enzymes that are very, very important for you to know. So if they ask you to talk about the bypass enzyme, remember, you have glucose-6-phosphatase, you have fructose 1,6-bisphosphatase, you have pyruvate carboxylase, and you have what we call the malic enzymes. Don't forget, pyruvate to oxaloacetate inside the mitochondria. You are seeing it on your screen. Oxaloacetate forms what we call malate. Malate leaves to a shuttle system to the cytoplasm. The cytoplasm is converted to oxaloacetate. You are seeing it on your screen. Oxaloacetate forms what? PEP. So when you form PEP bypass step 7 to step 5 and be in step 4. Step 4, after step 4, fructose 1,6-bisphosphate bypass step 3 and step 2, and it forms glucose-6-phosphate. Glucose-6-phosphate now form glucose. So I hope it all made sense to you and you are understanding everything I've talked about. I talked about it many times. So pyruvate is the key substrate in gluconeogenesis here because most people forms it. I get it now. So pyruvate carboxylase is very important because anything carboxylase needs what? Biotin, vitamin B7. So vitamin B7 is needed for this reaction 100%. They need it very well for this enzyme 100%. But oxaloacetate for me, pyruvate to what we call the malic enzymes. Don't forget. So you also need ATP, ADP, NAD plus, and all of them are needed in this question. They are needed very well here. So hope you understood, and you understood very well. You understood all these things I've talked about. I've recited it for you. You can get it to you can get gluconeogenesis to lactate, to anaerobic glycolysis. When there's high amount of lactate, Cori cycle help you to reverse it back. You can get it to glucose-alanine cycle, whereby your muscles will be broken down to form glucose. Your muscles will be broken down through the process of what we call transamination. Also, you can get it to beta oxidation. So in beta oxidation, you form succinyl-CoA, and when you form succinyl-CoA, from there you can form malate. From malate leaves do the same reaction. Also, you get it now, you can form it two malic enzymes too directly. So hope you understood all these things. Okay, now, so if you are just new to my channel, make sure to subscribe and turn on your post notification button so that anytime I drop any video, you'll be sure to be notified. If you want me to make a video for you, drop it down in the comment section. So you can also follow all my social media handle at MedGenius Academy in all my social media. Okay, now, so keep watching me, keep following me every day, every time. Bye bye.